Great. Thank you for the opportunity to talk a little bit about the roles of drug resistance and evolution and sort of thinking about these dynamic concepts. And today I'm going to talk more about a conceptual framework to sort of try to think about some of the considerations around where key knowledge gaps are present in this subject area. So we've been using drugs for a long time in treatment of malaria, and resistance essentially occurs in every place where antimalarial drugs have been deployed. Antimalarial resistance occurs essentially to every drug that's been used to treat malaria. But keep in mind that many of these antimalarial drugs still remain effective, and I think that's something that we have to keep in mind as we talk about drug resistance. We tend to use antimalarial drugs in combinations to reduce this risk of drug resistance and protect those drugs for use. But we do see, for example, drug resistance to artemisinin, partner drugs like piperaquine emerging in increasing frequency. And I show you a picture here from Mioto et al., sort of looking at some of the Kelch13 mutations that are associated with artemisinin resistance in Southeast Asia. So it's really important that we do surveillance for drug resistance as we're looking towards changes and moving towards elimination. We now have been adding, on top of this use of drugs to treat malaria, we've been adding added pressure for drug on parasite populations, largely in the context of trying to move towards elimination. So we've been doing things where we're increasingly using these antimalarial drugs. They're directed at the population level. And sometimes these are being delivered without really knowledge or need to look at infection status in the individual. So the overall goals here are to really reduce and potentially interrupt transmission, focusing on trying to get rid of parasites from infected individuals and from reservoirs that may not be identifiable under previous sort of strategies. The strategies that involve use of drugs may be preventative, so things like seasonal malaria chemoprevention or IPTP, or they may be strategies that are directed at, let's do more active detection and treatment, like a mass test and treat or screen and treat approach, or let's try to really reduce the reservoir, so a mass drug administration. But concerns include potential rise of drug resistance, and also as we reduce populations, potentially there's waning immunity and the impact that that may have both on parasite infections and also drug resistance. So in terms of some of these drug-based strategies, again, we're looking at applying these to whole populations. So a larger proportion of the parasite population is subject to drug pressure. And again, history tells us that drug pressure will select for variants that can escape this pressure. So there's this need to really be sure that we're looking at what's happening in terms of drug resistance, alleles and frequencies and phenotypes in the context of these campaigns. Reducing these parasite populations using these drug-based interventions, therefore, may result in sort of a selection of drug resistance variants. So again, looking at both changes in evolution in the parasite population structure are going to be very important, and keeping our eye to the mind that this population of parasites is changing, is dynamic. And so what we've been applying and working and making decrements in terms of transmission may in fact start to become undermined as those interventions, drug-based interventions, may now contend with a parasite population that's largely resistant or refractory to those interventions. So this is just a strategy from PATH looking at sort of mass test and treat. The idea is you identify people and treat them, the individual who's infected. A focal mass drug administration, whereby household, if anyone in the household is positive, the household is treated. Or mass drug administration, where we don't look for who's infected or not, we treat everyone in the community. So this is getting to this idea of evolution and population bottlenecks. And it's really this idea of trying to essentially crash populations to really reduce parasite populations. And then, again, the risk of potential selection for survivors. So this is based on evolutionary principles that as we massively reduce populations, we have fewer variants. So all these colors over here, with time we have fewer variants. And if we're using drug to kind of reduce this population, history will tell us that the likelihood that we're going to select variants that are able to survive this is increased. And so appreciate that a lot of these approaches are to crash to zero, but survivors have the opportunity potentially to rise in frequency. And again, it'll be very important to think about what are the critical inflection points or thresholds between this population decline and potential selection for variants for drug resistance. So you've seen this slide before, but thinking now in the context of a drug-based approach, we have a lot of natural kind of bottlenecks. We have natural bottlenecks within the life cycle of the parasite, and I'll point out a few in a moment. But I also want to remind you that we have natural bottlenecks in the population-based, if there's seasonal malaria transmission, we have dry season, we have other kind of dynamics that are also important to consider. So with drugs, maybe it's important to think about targeting specific life cycle stages or processes. So perhaps as we get to low levels, the moment of invasion is a really important place to be looking at. Maybe it's important with regard to transmission, for example, to look at specific life cycle stages like gametocytes that really have a tremendous potential or impact on transmission. And again, thinking about, as we're applying drugs, the possibility that parasites may hide out or sequester or be sort of reservoirs within the human, and how that may also potentially impact emergence and selection for drug resistance. So I'll give you two examples from history to think about in terms of population structure and changes in population structure and its impact on drug resistance, and also on looking at different types of variants, genetic variants. So history has shown us that, for example, as we've applied chloroquine pressure, this is it selects for and maintains mutant PFCRT parasites. But parasites that have this mutant PFCRT have a fitness cost. So if that drug pressure is removed or reduced, and we see this in Africa, because there's a large population structure with wild type versions of that allele, and there's the opportunity for outcrossing, and again combined with this fitness disadvantage, we can see repopulation of wild type alleles within African parasite populations, for example, where transmission levels are relatively high. In contrast, in populations where there's been sort of low transmission and a lot of application of drugs, we now see generally that the PFCRT version, the mutant version, is really almost fixed in a lot of these populations. So again, there's no wild type population to be able to outcross to, and so we see different frequencies of alleles depending upon transmission level and population structure, and also the importance of fitness and how that plays a role in these dynamics. The other thing to consider is the type of variant. So when we think about drug resistance, if we think about a genetic basis, we have to think about, is it one or more than one mutation that's required? We have to think about, is it a single nucleotide polymorphism or a copy number variant? They have different dynamics and heritability. And we also may have to think about non-genetic or epigenetic kind of considerations for drug resistance. But thinking about these sorts of changes, just looking at SNPs like PFCRT or PFKelch13 versus copy number variants like we've seen in PFMDR1 and now the plasmepsin 2-3 copy number variants associated with piperacin resistance. So drug pressure may select at different rates these kinds of variants, and sometimes it's maybe required that we actually keep pressure on a population, say for copy number variation, in order to see particular variants. So those will also play a role in the selection for and persistence of drug resistance in populations based on evolution. So another thing that's been largely looked at in the laboratory but also has implications for the field is, again, using evolution and our understanding of evolution around how drug resistance emerges and the ways that we can kind of almost take advantage of that. So lab studies have identified drugs with sort of inverse effects on specific targets. One drug is going to, push, if you look at this resistance here, we get resistance to drug A, but that makes that parasite susceptible to drug B. Now we can put drug B on the population, and it converts it to another alternate allelic form, which actually is now susceptible again to the alternate drug. So these drug combinations might be a way of either reducing emergence to resistance or preventing it at all, sort of a resistance to resistance. And so, again, we're starting to look at what we think about as smart combinations that might suppress resistance. And I'll talk more about that and the field implications in a little bit. So I'm going to leave you with just some thoughts about knowledge gaps and things to think about. And I know we're going to have a panel discussion to consider some of these. But I think one of the things that we need to think about is the role of the parasite reservoir. And that reservoir can be twofold. One is the parasite reservoir in a population that's not maybe susceptible to or accessing drug pressure. And the other is to think about within host reservoirs that may be differentially accessing drug that is provided to an individual. We have to think about the role of fitness in drug resistance. And there's good evidence to suggest that fitness costs for drug resistance alleles, again, may be something we can leverage or take advantage of. We have to think about the mechanism of resistance. Do we have one or more genetic mutations, or is it epigenetic? Do we have SNPs or copy number variants? How is that considered in terms of the emergence and the spread of that resistance? So that's kind of looking at the stability of the variant. We also need to think about the life cycle stages, what drugs can target and have the greatest impact. If our goal is moving towards elimination, how can we interrupt transmission using drugs, and what are the consequences for that on drug resistance? I think it's very important to consider these two dynamics, the transmission level changes and the potential rise, the sort of evolutionary arms race that I talked about earlier, where we sort of have these trade-offs between population structure and also potential rise of resistance. So how does the level of transmission impact the likelihood of emergence? And then looking at that transmission level or even patterns of transmission. So how do we consider superinfection and co-transmission and the bringing in of alleles? Does that contribute to or accelerate drug resistance? We have a lot of learning to do about that. And then thinking about losses of immunity as we lower population sort of exposure and how the human immune system participates in this, ways that drugs are able to work and resistance. And also considering different parasite species. So I think a lot often about Plasmodium falciparum, but the dynamics change for these other species and the way that the drugs act on them as well. Thinking about smart drug combinations, how can we limit that emergence? And I think we really need to think about how these may be deployed. People talk about, should we do them simultaneously? Should we do them sequentially? And there's some potential trade-offs here, especially as lots of drugs are available in many places. We may have some problems, with, say, sequential, if we are trying to drive a susceptible population by one drug combination, if the alternate sort of drugs are available and adding and keeping pressure on that parasite population. So those are things to consider, and I will just end by acknowledging a lot of people in the Harvard Broad kind of team, a couple of photos, Dauda Njai, key collaborator in Senegal, some of our team in snowy Boston, and then the workshop group, and a lot of collaborators that we work with and participate in the thinking about these ideas. Thank you.